protein we take because some amount of protein we also lose every day. So that loss is to be compensated by somehow, and that is to be maintained from an external source, particularly essential amino acids also. Without that we cannot sustain, and that's why protein requirement in the diet is less, 10 to 15%, not more than that, because we basically compensate what we have lost. Now, there is constant turnover of protein. Turnover means there is constant breakdown of protein in the body in diverse forms from various tissues, and then it is also regenerated at the same time, means synthesis is going on. So, synthesis and breakdown are going on simultaneously, so that there is no net protein loss from the body. Now, when somebody is growing, when the child is growing, or a pregnant mother is having a baby within her womb, practically the amount of protein which is to be taken by a growing child or by a pregnant woman or by a convalescent patient, particularly means those who have recovered from an illness where she has lost some amount of protein in the disease, they require an additional amount of protein. Why? When the body is in equilibrium, that is in a steady state, when the loss and intake and output are same, that is body is in net nitrogen balance. Now, when the loss is more, loss in terms of nitrogen, because amino acid contains nitrogen, almost 16% it is a fixed part. So, that excretion of amino nitrogen as ammonia is an indicator of loss of nitrogen. So, when somebody is losing the output of nitrogen is less than the intake, then the body is in the positive nitrogen balance. Means some muscle has grown, the baby is growing, or a pregnant mother is having a fetus which is also developing, so he is getting some amount of weight that is positive. The intake is more than the output, then that is called a positive nitrogen balance. And the intake, the output is more, particularly when somebody is suffering from some disease where protein breakdowns are going on. That is called a negative nitrogen balance. Anyway, in any situation of protein breakdown, normally it is going on. So, during that breakdown procedure, at the end, just like digestion, there will be a breakdown of proteins in the body, in the cells, in all cells. And at the end, you will get an amino acid. That amino acid will be further broken down, and there is an amine group. That amine group will be releasing an ammonia, and there will be carbon skeleton left. So RCOH NH2, that formula you know very well about the amino acid. I don't want to repeat again. So once I want to say that this is the structure of for all amino acids. So what we get when the amino acids are finally broken down, it is deaminated, and once it is deaminated, you will get this as ammonia. And the remaining carbon skeleton part is there. So both are used. So this ammonia is used to convert a keto acid into an amino acid. Okay. So from some non-essential amino acids can be formed from this ammonia, and ammonia can be used for other biomolecules, and the carbon skeleton will be used for some other purposes. So normally there is some amount of ammonia which is present in our body, in everybody's body. But when it reaches a pathological state, when the levels are very high, that is called hyperammonemia, and that is toxic. That is toxic to the brain. So therefore, body has an extensive mechanism not to allow hyperammonemia at any cost. It traps the nitrogen the moment it is formed. There is no storage organ of nitrogen, but nitrogen remains trapped within the molecule. It is transferred from one molecule to another molecule, and that molecule circulates in the body from one organ to another organ. That's it. So ammonia is trapped. Now, why ammonia is toxic? That is a big question. So I have written an answer. So you can note down why ammonia is toxic. Summary I have written. In the examination you have to write, there is a time constraint. So that's why I have made this thing as little as possible, but you can modify it in your own language. So first part I am telling you, just try to understand, then try to understand this part. Don't write, I will give the time. Each point I am discussing. First point, there are 4 points, okay? These 4 points you must remember and try to understand first. So first point is, write down now, write down. Uses or depletion of alpha ketoglutarate to form glutamate. Why ammonia is toxic? The answer to that question are basically 4. This 4 point is usage or write down a little bit faster. That is the only point you have to note down. Rest you will get from some other books. If required, I will instruct you this to note down. So there, see, alpha ketoglutarate, if you remember correctly, the TCA cycle, alpha ketoglutarate is an intermediate product. And if alpha ketoglutarate is taken up from the TCA cycle will not operate completely. So energy depletion will be there. The cell will be deprived of energy, and brain cells require continuous energy, and all body cells. So impairment of TCA cycle in neurons, so energy production will be decreased. You can write down in your own way. Ammonia reacts with alpha ketoglutarate to form glutamate. So depletion of alpha ketoglutarate, so impaired function of TCA cycle in neurons, depletion of energy in neurons. That is the simplest way of expression. I mean, you can modify the answer in your own language. Do you understand? All of you understood this thing. So alpha ketoglutarate is taken up from the TCA cycle. So energy production is decreased. Second, depletion of glutamate required for inhibitory transmitter gamma aminobutyric acid for glutamate formation. So GABA production is decreased. GABA production, glutamate is used for glutamine formation. So depletion of glutamate means glutamate is required for gamma aminobutyric synthesis, acid synthesis. So therefore, a depletion of inhibitory neurotransmitter. GABA is an inhibitory neurotransmitter, means it inhibits the activity of the brain. So when the brain is excited, so we become very much overactive. When the brain is depressed, everything gets depressed. Like respiration, heart rate, even our mental flowing, everything will be depressed. The alertness, whatever alert we are aware, that part, the brain will not function properly. So that is an inhibitory neurotransmitter. So that secretion is decreased. So then excess glutamine formation. Glutamine is an osmotically active substance. So when something is accumulating within the cell, which is osmotically active, that cell will draw water. So that causes brain astrocytes to swell up. There is one type of cell that is called astrocyte. So astrocyte will swell up, there will be cerebral edema, the swelling of the body, swelling of the brain, not body. And because of the swelling of the body, there will be increased intracranial tension. And since when the brain intracranial tension means brain again will not function properly and there will be disturbance. And second point is increased glutamine is an excitatory neurotransmitter. So it will excite the brain and brain cell will die because of excitotoxicity. So there is a NMDA receptor, that is N-methyl D-aspartate receptors. This receptor will be activated and there will be excitotoxicity. So now I am clarifying, astrocyte contain most of the total brain glutamate synthase. Glutamine is produced from glutamate. Glutamine is produced from glutamate using glutamine synthase enzyme. While all neurons contain virtually all brain glutaminase. Excess glutaminase leads to osmotic imbalance of astrocytes, as I told you. Water intake swells up the astrocyte leading to cerebral edema and that causes raised intracranial tension. Third point, excess tryptophan, ক্যামেরাটি বন্ধ করো, লেখো, খাতাতে লিখতে হবে, পরীক্ষার খাতায়, এগুলো লিখবে ভালো করে। Excess tryptophan entering into the neuronal cell in exchange of glutamine, producing excess excitatory neurotransmitter serotonin. Because tryptophan basically enters into the brain, excess glutamine comes out from the cell in exchange for amino acid tryptophan, and excess tryptophan leads to production of serotonin, which is an excitatory neurotransmitter. So now all three, this GABA production is decreased, means inhibitory neurotransmitter is decreased, means there will be stimulation. Then excess tryptophan is called excitatory stimulatory, and also that this part glutamine. So this is these all 4 points are the causes of hyperammonemia. So clinically what you will get all neurological disturbances. Say there will be confusion, disorientation. I mean, this cannot be told in a class, you have to see a patient, then only you will see. All these things will not be present at a time. But in a full-blown case, you will get it, everything. Means when the patient is almost about to die, because confusion will be there, disorientation, lethargy, tremor, seizures, respiratory alkalosis, cerebral edema is there. So diverse thing will happen. Ultimately, patient will go to coma, means unconsciousness, and then if left untreated, or if the patient doesn't respond to treatment, that will lead to death also. So there is slight amount of gastrointestinal vomiting or loss of appetite will be there, but mostly the features of hyperammonemia are neurological. Slurring of speech will be initial stage. So treatment, since there is a clinical aspect, so this type of patient will come. Now how will you treat? Now you have to identify what are the causes, what is the underlying cause in this particular patient. What has happened, what has gone wrong. In most cases, in adult stages, it is due to liver failure, because liver is the organ where ammonia is detoxified into urea. So that organ will be at fault. You will read at the end of that the urea cycle will be complete, you will understand better. So ammonia, which is toxic, is detoxified to urea in the liver. Now, if the liver is damaged to a significant extent, which will be unable to detoxify ammonia into urea, then the ammonia level will start rising. And then when it will be severe, then it will cause brain damage. And if it goes on continuing the situation like that, liver cells are going, damage is going on, and ammonia is building up in the, ammonia level in the blood is built up. So that will cause damage and that will cause hyperammonemia. So underlying disease condition you have to treat. Or there might be a condition where the urea cycle enzymes, which I will teach tomorrow, is any or one or more than one enzymes are defective, then urea cycle will not operate. So that will be found in the pediatric group, newborn, or in the early childhood, not in the adult patients. So treatment mode will be different based on the underlying cause. Second part is that dietary changes. Normally, as I said you that this is from source, main source is from amino acid. So dietary proteins is to be restricted. That was the dictum earlier. Now this is not emphasized much because by the time the patient comes in a very poor condition, in a poor health condition, when not much protein restriction is done, which was taught during our time. So lactulose is given. So one source of ammonia in the blood is bacteria from normal gut flora of the intestine. I will discuss in a short while. I am telling you in advance. At the end I will tell you once more so that you understand better. We get some amount of ammonia from intestinal bacteria. So that part has to be, somehow we have to stop having ammonia from intestine. That is why lactulose is given. It is an orally non-absorbable disaccharide. It decreases production of ammonia and absorption of ammonia by the intestine. Now this gut flora which is maintaining, which is producing the ammonia, our normal flora as well as pathogenic bacteria if it is there, now that need to be destroyed. So we usually give an oral antibiotic which will destroy the intestinal organisms, microorganisms, so that they will be destroyed. So they will no longer be able to produce ammonia. So one source of ammonia will be produced, so that will control somehow ammonia production. Then since brain is having edema, so brain cells are swelled up, have all swelled up. So from that brain cell size has to be reduced to normal. So something has to be given extra, hypertonic saline if it is given. So that will exert an osmotic pressure again on itself and it will draw water from the brain or any body tissue. Particularly in our time mannitol used to be given. Now nobody, I have used also mannitol. Instead of mannitol, hypertonic saline is used. And L-ornithine, L-aspartate is given in hepatic encephalopathy in the adult. But if it is in a urea cycle enzyme defect, then L-carnitine, L-arginine, and finally when everything is over, the last stage will be almost hemodialysis to get rid of ammonia. So this is a brief outline. You need not note down everything, but I have given a brief outline why these are, what is the clinical aspect of hyperammonemia. So that you will learn in detail in your 5th year class. This is not actually the purpose of today's class. Today we want to discuss, since you see here, describe the formation, transport, detoxification of ammonia, ammonia toxicity and its clinical significance. What I did the last part first, so that you understand the other parts. So ammonia toxicity and its clinical significance I have explained. Now I will come back to formation. As I told you that ammonia production is done by all tissues, because all tissues these amino acids are there, and they produce ammonia, and the ammonia is finally transported to the liver for detoxification. So various sources of ammonia in the body. One is amino acid, that is dietary amino acid, protein-rich dietary amino acid. If you take excess amount of protein, then that will cause high ammonia level, and then that high ammonia level will be detoxified by the urea and it will be cleared in the urine. So, excess dietary intake does not have any role. Second part, biochemistry part, removal of amine group from amino acids, that is transamination followed by oxidative deamination. These two are coupled together, that is called transdeamination. This is note down. Removal of amine group from amino acid, transamination followed by oxidative deamination, and there are 4 other methods, 3 other more methods which are non-oxidative, that is reductive deamination, hydrolytic and intermolecular deamination. From glutamine, ammonia is produced, but as I told you, it remains trapped within the molecule. From kidney, ammonia is not released into the circulation. It is stored in the glutamine. Renal glutaminase is there, so it releases ammonia from glutamine to glutamate. It is formed and ammonia is released. Now, this ammonia is used for acid-base balance maintenance. This you will read later during acid-base balance. So, that is why ammonia is produced by the kidney, glutamine is stored as a stored form in the molecule. It is used as an acid-base balance maintenance of the pH of the body. That is it. So, intestine there is an intestinal glutaminase, and in the brain also there is a glutaminase. Bacterial action in the intestine, urea splitting bacteria are there. So, they produce urea. Urea, what is urea? Urea splitting bacteria. So, 2 molecules of ammonia will be produced, and that is done by the intestine. Normal flora is there, they produces ammonia. Another source is amines, ammonia. Biogenic amines we get from diet, polyamines. Any structure in the body which is having a nitrogen, like purine, like pyrimidine, they can also produce ammonia, but the amount of ammonia produced by these molecules are less. So, most important source is basically amino acid, and that is through transamination and deamination, oxidative deamination. These are 2 main sources. So, and another is L-amino acid oxidase. From L-amino acid, again you see amino acid is converted into ammonia. So, there are some minor pathways, but these pathways contribute very little ammonia to the blood. So, they are less important. So, how the ammonia is used by our body? Now, we have produced ammonia. Now, there is some amount of ammonia which will be gainfully used, like alpha ketoglutarate, it is taken up and it forms an glutamate. And from glutamate, other amino acids can be synthesized. L-amino acid, the glutamate can be formed. From glutamate to glutamine can also be formed. So, that glutamine will be used for, say, purine synthesis, nucleotide synthesis, amino sugars, tryptophan, histidine. So, as I told you, the amino acids, this group is used for making some new amino acids. Third part is aspartate to asparagine. One amino acid will be added, then aspartic acid will be converted to asparagine. Fourth one is most important part. For that, there will be dedicated class tomorrow, that is urea cycle. So, ammonia is mainly detoxified in the body as urea, and that is done by the urea cycle. So, most of the ammonia, major part of the ammonia are detoxified as urea by the liver, and this is that urea is a soluble one, it is a non-toxic product, and that is excreted through the urine. Okay. Rest other, there are, some important functions where the ammonia can be used gainfully, not as a waste product, are these. So, deamination, remember deamination means you are getting rid of ammonia group, deamination. There is no more amine group here. So, amine group is to be removed, that is a deamination. So, it can be done mainly by oxidative deamination. Remember, note down. 4 types are there, but the oxidative deamination is the most important part. Just for information sake, we write down there are non-oxidative part also, because when you answer very well, the examiner might ask you, tell me something about non-oxidative. Is there other pathway? Then the non-oxidative part, there are reductive, hydrolytic, but an intermolecular. First you know, concentrate on this one. This is the most important part, that is L-glutamate dehydrogenase is involved. What is done here? You see glutamate is here. Glutamate means it is an amino acid, it is having an amine group. Now glutamic acid is having 2 hydrogen. Now it is getting rid of that hydrogen. So NADH is formed, and it is formed in alpha ketoglutarate. So the concerned enzyme is L-glutamate dehydrogenase. Basically here there is a deamination, but it is coupled with 2 oxidation also. So that is the reason why it is called an oxidative deamination. Is this part clear? Here the glutamic acid is having extra 2 hydrogen here, you see. NADH is acting as NA acceptor, NADH. These 2 hydrogen is getting rid of. So glutamate is basically getting converted into alpha ketoglutarate. So there is an oxidation, and this oxidation is coupled with deamination. That's why its name is oxidative deamination. Okay. So this is the most important pathway, and it plays a central role. It is, this enzyme is present maximally in mitochondria. You see at this stage, you remember it is present in the mitochondria. So that deamination, oxidative deamination will take place in the mitochondria, not in the cytosol. In contrast, transamination occurs in the cytosol, not in mitochondria. So this reductive deamination, what happens? You see, reductive, there is reduction, but amino acid is getting converted into fatty acid. There is hydrolytic deamination, means hydrolysis is there, so a hydroxy acid is formed. And intramolecular deamination, and amino acid is converted into an unsaturated fatty acid. Okay. So something else is produced from here through non-oxidative pathway, which are minor pathways, not the major pathways. Major pathway is this one. So L-glutamate dehydrogenase is a little bit information about this thing. It is a zinc containing metal enzyme. So when the body energy is adequate, cellular energy is very much adequate, then this cycle doesn't operate. Then the protein doesn't break down normally. In a starvation condition, basically at the extreme stage, after the glycogen storage and the fat stores are depleted, then only protein is broken down. So when there is an energy excess, when that means ATP, GTP and NADH, they will act as an allosteric inhibitor. They will not allow to operate that deoxidative deamination path. Similarly, when the body is in need of energy, the ADP, GTP, they will act as an allosteric activator. So low levels of energy means decreased ATP, so increased catabolism of amino acid will be required. Only in that case that amino acid will be activated. So metabolic significance is that it is a reversible reaction. means both anabolic and catabolic. It channels, when the catabolic action is there, it channels nitrogen from glutamate to urea. When there is anabolism, means nitrogen has to be stored. Here nitrogen is, we are trying to get rid of the excess amount of nitrogen. So that's why it is catabolic. It channels nitrogen from glutamate to urea. Alpha ketoglutamate getting converted into alpha ketoglutarate, ammonia is released, excess ammonia is getting converted. Anabolism, now amination of alpha ketoglutarate by ammonia to form glutamate. Now we need to synthesize ammonia. So transamination means it is a transfer of amino group from an alpha amino acid to a alpha keto acid. In the process, what happens? One alpha amino acid is involved, one alpha keto acid is involved. Now the amino group which is donating that amino acid, this is acting as an acceptor. So that this amino acid is getting converted into a keto acid, and this keto acid is getting converted into an amino acid, and this is interconvertible. So transamination reactions are interconvertible. It is called aminotransferase, as there are two important aminotransferase or transaminases, aminotransferase and transaminases. These are both synonymous. Aminotransferase means you are transferring amino group to something else, and transaminase means you are transferring amino group to something else. When we were a student, this terminology was not there, aminotransferase term was not there. It was transaminase only. When I studied MD, then only, I mean in the late 80s, then I came to know about that aminotransferase term. Okay. So it is basically the same thing as you will see in an example, I will tell you. So you write down some salient features. It transfers amino nitrogen to alpha ketoglutarate to glutamate every time. Every time basically alpha ketoglutarate is this part, then I will explain one. Transfer one amino nitrogen to an alpha ketoglutarate to form glutamate, and it is a free reversible reaction, as I told you. It is used as an amino acid biosynthesis. And each aminotransferase is specific for one pair only. The same pair will not function for other. This last part, what I have written in the red, you note down. Alanine is a substrate for glutamate aminotransferase. The alpha amino nitrogen that undergo transamination is concentrated in glutamate. Because glutamate is the final, only undergo glutamate. Why glutamate is concentrated? Because glutamate is the only amino acid that undergoes oxidative deamination, and that happens in the mitochondria. And without that, urea cycle will not operate. That's why all amino acids are basically finally converted into glutamate for final excretion by the liver or as urea. Okay. So there are different amino acids you see here. Alanine aminotransferase. Alanine is there. Alpha ketoglutarate. Having an amino group. So alanine is transferring the amino group to alpha keto group. So alanine will be converted into pyruvic acid. when this reaction will proceed in this direction, alanine aminotransferase. Transaminases. Glutamate, pyruvate, transaminase. So glutamate, is this part clear? GPT never say. S means serum. It is present as an enzyme which we measure. When we measure in the blood serum, then it is called an SGOT, SGPT. But otherwise, GPT is synonymous with ALT. Now, instead of this thing, suppose this alanine here I have written like this, okay? Instead of alanine, we are writing aspartic acid. Okay? Alpha ketoglutarate. It will be converted to oxalic acid. Aspartic acid will be converted to oxalic acid and alpha ketoglutarate to glutamate. So when we write it is, this will be aspartate transaminase. When it is in this side, that is oxalic acid. So SGOT is equivalent to this is what is aminotransferases and transaminases, nothing more than that. Is this part clear? Now, this is having an importance because liver is having a high concentration. So, when liver cells are damaged, obviously the enzymes, this is another part I will tell you in detail. So, this is having a clinical implication. Now, here you look down here. So, as I told you, one example here, just consider here. You see, first reaction you look at. Now, alanine aminotransferase. First you concentrate here, alanine is there, alpha ketoglutarate is there. So, when the reaction will move in this direction, alanine transaminase, ALT, when it is moving in the other direction, that is glutamate pyruvate transaminase. Is this part clear? Here you see reaction. Could you understand? When the reaction is moving in this direction or moving in this direction, the enzyme is same. Name is different. The purpose, why it has been named so, the purpose I am telling you. It is a reversible reaction. So, just to remember, alanine, alpha ketoglutarate, only thing is that when this enzyme will operate, you have to write down this one. Which direction you are maintaining. I mean, you can write both the names, there will be no problem. But only thing is that what is getting converted, you have to know what you are using as a substrate. So, same thing what I have written here, aspartate transaminases. So, can you note down in both the cases, alpha ketoglutarate is involved and glutamate is formed. So, at the finally the amino group is concentrated in L-glutamate. So, mechanism of this transfer is basically a ping pong type of mechanism. Vitamin B6 is used as a pyridoxal phosphate is used as a cofactor or coenzyme you can say, vitamin as cofactor. So, in the first step, basically it binds with the pyridoxal phosphate and forms a pyridoxamine, and then that alpha amino group is converted into a keto group. That is Pong mechanism. Basically, nothing more than that. This is they have written. I am teaching you in the class. So, what is ping? This is ping. First substrate is binding. It is binding to an enzyme. Then this is an enzyme intermediate. Then product is formed. And another substrate is binding, and another product is formed. So, there are two substrate, two product that you have seen. So, that is the ping pong mechanism. That two substrate, two products. So, it is not two substrate, one product, or three substrate, one product. It is not like that. So, that's why it is named as a ping pong mechanism. Regarding transamination reaction, one point you note down, all the amino acids except lysine, threonine, purine and hydroxypurine participate in transamination reaction. এইটা পরে না পড়লে হবে। I told you this thing, pyruvate is converted to alanine, oxalic acid converted to aspartic acid, alpha ketoglutarate to glutamic acid. So, these amino acids which cannot be synthesized in this manner are therefore essential, something which we cannot produce in our body. Transdeamination is nothing, it is a term only, it is a coupling of transamination with deamination. Both the processes are coupled. One is occurring, transamination occurs in the cytosol, deamination occurs, oxidative deamination occurs in the mitochondria. And when both the systems are coupled, reactions are coupled, that is called oxidative transdeamination. Transamination followed by oxidative deamination. So, I told you repeatedly the L-glutamate dehydrogenase plays a central role in ammonia removal and nitrogen metabolism basically. So, transfer of this amine nitrogen to alpha ketoglutarate to form glutamate. Transamination takes place in the cytoplasm of the cell. Repeatedly I am telling, the final amino group is transferred to glutamate, which is deaminated in the mitochondria. Liver mitochondria contains glutamate dehydrogenase in the highest concentration. Write down. That is the reason why it happens in the urea cycle, operates in liver only, why not in other organ. Amino acids are deaminated at the rate of 50 to 70 grams per day. That's why we told you that we require protein, intake of 1 kg body weight. That is about a 60 kg adult will get an 60 gram of protein. So, glutamate is dehydrogenated in the final reaction. Now, how the ammonia is carried, transport. As I told you, the ammonia is transported as glutamate within the cell through the aminotransferase. And the glutamate dehydrogenase does just the opposite reaction, alpha ketoglutarate to glutamate, glutamine and alanine. In the muscle, alanine is produced. In the brain and intestine, glutamine is produced. In all other body tissue, glutamate is produced. So, ammonia is transported in the blood in 3 forms. From majority of the cells as glutamate, so from brain and intestine as glutamine, and from muscle as alanine. Because muscle does not have the glutaminase enzyme. So, this is how the ammonia is transported into the blood in 3 forms mainly, and there is no storage organ of ammonia, I repeat. So, ammonia transport, glutamic acid acts as a link between the amino group of amino acid and ammonia. Cellular ammonia is concentrated as glutamate because glutamate dehydrogenase will be required for oxidative deamination. All other amino acid, and that is the reason why the concentration of glutamate is almost 10 times higher than other amino acids. Because all the amino acids are present in the blood, in some conditions it is getting distributed. Glutamate is present in highest concentration because all the cells have transaminated into glutamate form. And as I told you, final transport form of ammonia to liver is in 3 forms. One is glutamate from the almost all cells, from the brain and intestine as glutamine, and from muscle as alanine. Because there will be a cycle called glucose-alanine cycle, perhaps it has been told. During starvation, that cycle operates. When basically somebody undergo starvation, means prolonged fasting, hepatic glycogenolysis, then muscle glycogenolysis, hepatic glycogenolysis, muscle glycogenolysis takes place. Then fatty acid breakdown is done. Then from the muscle, it will act as a source of glucose, but not directly, through alanine. So glucose-alanine cycle will operate. From the alanine, glucose will be produced. So now you try to understand, you have understood why we have given. This is the last slide for today's class. As you can understand that underlying cause of hyperammonemia, you have to treat what is the real cause of the disease. Now dietary changes, why we are restricting protein, because that is a source of ammonia. So if required, if the patient is really requires ammonia protein restriction, then only you do it. Otherwise, you don't do it. Because patient condition by that time is very bad. Lactulose is given in the intestine to prevent intestinal ammonia production and transport. Rifaximin is given to cause death of the normal bacteria present in the intestine, so that they cannot produce antibiotic. Hypersonic saline is given because we want to reduce cerebral edema. And L-ornithine and L-aspartate is given in hepatic encephalopathy to stimulate ammonia metabolism. L-carnitine in arginine, tomorrow I will tell you in the class, the urea cycle defect and hemodialysis.